Good morning, everyone. Thank you for joining us for Winship Grand Rounds this morning. If you are an Emory University or healthcare employee and would like to receive CME credit for attending today, the access code is 353806 and can be found in the chat feature at the bottom of your screen for our virtual attendees. If you have any issues with this webinar or the CME login, please send Margaret Johns an email or drop a note via the chat feature. This morning, we are very pleased to welcome Dr. Timothy Gershon. Dr. Gershon received his MD and PhD in biological sciences from Columbia University and completed his residency training in pediatrics and neurology, along with a fellowship in pediatric neurology at New York Presbyterian Hospital in New York City. He also completed a fellowship in neuro-oncology at Memorial Sloan Kettering Cancer Center. Dr. Gershon currently is a professor in the Department of Pediatrics at Emory University School of Medicine. He serves as the director of the Children's Center for Neurosciences Research. He is a member of the Cell and Molecular Biology Research Program at Winship Cancer Institute at Emory University. He holds professional memberships with the Society of Neuro-Oncology, Child Neurology Society, American Association for Cancer Research, and American Neurology Association. Dr. Gershon's lab studies the interrelation between normal brain growth and pediatric brain tumor formation. His focus is on the cerebellum, which is a key site, as we know, of postnatal brain growth, and on medulloblastoma, a cerebellar tumor that is the most common malignant brain tumor in children. The lab works primarily with in vivo models and tumor metabolism, cellular heterogeneity, and microenvironment. His lab is studying therapies that target tumor metabolism, that block tumor recurrence, and that use the immune system for anti-tumor effect. The goal is to develop new treatments for brain tumors based on their unique biology. Please welcome Dr. Gershon today for his talk, Harmonizing Innate and Adaptive Immunotherapy for Brain Tumor Treatment. Thanks. Welcome.
Thank you for that lovely introduction, and thanks for the opportunity to present this talk. Thanks everyone for tuning in. All right, so I'm going to talk about immunotherapy for brain tumors. We're mainly going to be focusing on medulloblastoma, but what I have to say I think will transfer nicely to other contexts as well. All right, so what is medulloblastoma? Medulloblastoma, as Kimberly said, is the most common malignant brain tumor in children. It's a tumor of the cerebellum. Treatment for medulloblastoma was arrived at empirically. It's pretty effective. Thank you. It's pretty effective, but it kind of developed without any particularly rational direction. Treatment began with surgery. So in the 1920s, surgeons first became able to take these tumors out and have the patient recover. And this was immediately frustrating because the tumors always recurred somewhere else and still killed the patient. Things turned around in the 1950s with the advent of craniospinal radiation. So you could take out the tumor, but it would come back. You could radiate the place you took it out of. It came back somewhere else. A woman named Edith Patterson was the first author on the first paper describing craniospinal radiation for medulloblastoma. I like to say her name because it otherwise gets lost to history. She published this paper in 1953, and she took a disease that was 100% incurable and turned it into a disease that more than half the patients survived long term, which is great. But to think about how long ago that advance was, in 1953, How Much Is That Doggy in the Window was the top hit in the country. Chemotherapy was added since the 1950s in several slow clinical trials. Eventually, a regimen was found in the 1980s that actually improved therapy beyond the 50 to 60% long-term survival, getting us up to where we are now with about 80% long-term survival. Again, I put the top musical hit of the year just to give a sense of how long ago this development happened. So things have crystallized with about 80% long-term survival with surgery, radiation, and chemotherapy, and a consistent 10 to 20% recurrence rate. Recurrence is incurable. So for the people with recurrent disease, it remains 100% fatal. And survivors of surgery, radiation, and chemotherapy are at risk for significant long-term toxicities of therapy that can be disabling. Additionally, infants with medulloblastoma can't receive radiotherapy, and as a result, the recurrence rate is higher for them. Clearly, improvement is needed, but giving higher doses of radiation therapy in 15 different ways has been tried and doesn't produce improved survival rates. And giving higher doses of chemo, stem cell rescue, all kinds of different schemes of chemo have been tried. And again, the general finding has been that intensification doesn't produce reduced recurrence rate and doesn't treat recurrence. So I submit to you that radiation and chemotherapy are at the limits of their potential for this disease. That we're not going to get further with more radiation or more chemotherapy. And then the question is, what can we do to improve treatment, both to reduce toxicity for people who are getting cured and to actually bring about cures for people who are currently incurable. So new approaches are needed. And if we think about it, radiation and chemotherapy are targeting the tumor cells. And I submit to you, it may be time to think about targeting some other cell types. So what are the cell types in medulloblastoma? Again, going back to the 1920s when surgeons could first do resections on these tumors, we learned this is a tumor of brain progenitors. Harvey Cushing and his resident Bailey described a small round blue cell tumor that was frequently found in the cerebellum in children. They named it medulloblastoma. This isn't one of their slides, but this is an H&E from an actual medulloblastoma patient. You can see it's a hypercellular tumor. If you look at normal brain, you don't see the brain filled with cells packed in so densely. And in addition, there's certain key features. It's a small round blue cell tumor. The tumor cells themselves have very little cytoplasm. They arrange themselves in these kind of circular formations around eosinophilic areas. These are Homer-right rosettes. And what these are actually neural progenitors arranging themselves around areas of axons. So they're trying to do some sort of developmental program. They just don't do it right. Okay, radiation chemotherapy targets cells that are vulnerable to DNA damage, and that's how they work. We know that these tumors are heterogeneous. What if we target a different cell type in the tumor? Is this a crazy idea? Well, we've for a long time been using Avastin for brain tumor therapy. And if you think about it, Avastin is targeting the tumor cell, a cell in the tumor that's not a tumor cell. It's targeting the endothelial cells. This actually produces some beneficial effects. So the idea is not crazy, but what about targeting a different type of cell? And here's where I want to raise the fact that 10% of the cells in medulloblastoma are actually myeloid cells. This is data from a single-cell RNA-seq paper on medulloblastomas resected from patients that I was a co-author on. And we looked in this paper at cellular heterogeneity, but also heterogeneity between different patients. Now, medulloblastoma is a heterogeneous disease. There are 4 major molecular subgroups of medulloblastoma. Individual patients are all combined here with their cells. What we see is that patients with sonic hedgehog-derived medulloblastoma have their cells fall into a bunch of clusters. Patients with medulloblastoma, there weren't so many cells or so many patients, so we have only one cluster. The other groups, all these different clusters, patients with similar types of medulloblastoma cluster together. But the main thing that I want to show you here is the myeloid cluster is common to every single subgroup of medulloblastoma. And all the patients have myeloid cells that co-cluster together. This is true of other key stromal groups as well. Do these stromal cells contribute or play a role in tumor growth? Let's talk a bit about what myeloid cells are. So myeloid cells, to me, means 2 different groups of cells. There are microglia. These are resident brain myeloid cells. And there are macrophages, which are or monocytes. These are circulating myeloid cells that can enter the brain. Both myeloids, microglia and macrophages can be M1 inflammatory or M2 anti-inflammatory. If there are myeloid biologists in the audience, they will immediately think that this is overly reductive and simplified, but we're going to use this as a working definition. And if you work with me here, you'll see that we could divide this into kind of 4 possibilities. Microglia can be M1 or M2. Macrophages can be M1 or M2. Okay. Does this matter? Well, a couple of years ago, for example, an excellent paper came out showing that astrocytes within medulloblastoma are actually of tumor lineage, at least some of them, and that they signal to myeloid cells in the tumor microenvironment, and that cause these myeloid cells to secrete IGF-1. IGF-1 is an important growth factor in medulloblastoma, and if you block this signaling, it actually greatly slows tumor growth, at least in animal models. Okay, so work showing that these myeloid cells have a tumor-promoting effect. At the same time, a paper from my colleague here at Emory, Anna Kenny, and other investigators here, showed that there's a subgroup of myeloid cells with an anti-tumor effect. Specifically, a subgroup of macrophage monocyte myeloid cells that express the protein CCR2. This CCR2 positive macrophages have an anti-tumor effect. Okay, so how does this work? We have pro-tumor, we have anti-tumor, and again, there's 4 different kinds. So I submit to you, myeloid cells can be both tumor promoting or tumor suppressive. And then the question is, can we find a therapy that turns off the tumor promoting function and encourages the tumor suppressive function? So that's the hypothesis I want to present to you. Stimulating the innate immune function of myeloid cells will actually switch them from tumor promoting to tumor suppressive. To test this, we need an animal model, so I'll very quickly introduce that. So here we have a section of a mouse brain in sagittal section. This is a 7-day-old mouse. This mouse was injected with EDU the day before it was harvested, which is something we can stain red to show where there are progenitors in the brain. It's a DNA base analog that we can stain. And what you see is there's a region in the brain, towards the front of the brain, with a lot of neural progenitor proliferation. This is called the rostral migratory stream in the subventricular zone. And this is going to contribute olfactory neurons throughout the life of the animal. Additionally, there's this area in the back, the cerebellum, and all around the outside of the cerebellum is intense neural progenitor proliferation. There's another area of postnatal neurogenesis in the brain called the hippocampus. At this low magnification, you can barely see it, and this is because the amount of proliferation in the hippocampus is so much less than in the cerebellum or the SVZ that we just don't see it when we look out at this low mag. So this is, I mention all of this to say that there's an intense amount of postnatal progenitor proliferation going on in the cerebellum. And this proliferation is driven by a specific mitogenic signal called sonic hedgehog. Activating mutations in the sonic hedgehog pathway induce medulloblastoma in patients, and about a third of the patients have either an activating mutation or other evidence of sonic hedgehog pathway activation. In addition, we can introduce these mutations into mice and recapitulate medulloblastoma formation endogenously, as we see in this example of medulloblastoma, human brain, similar mutation introduced into mouse. You get a tumor in the same region. Okay, the model we use depends on smoothen. Smoothen is part of the sonic hedgehog receptor complex, and we have a constitutively active allele of smoothen. It's on a lock-stop-locks configuration. This is important because we don't want the breeders to be developing brain tumors. We want to be able to breed mice where the pups will get brain tumors. If we cross one of these mice with a mutant smoothened on a lock-stop-locks, we call it SMO-M2 mouse, with MAF1 Cre mouse that expresses Cre in the cerebellum, we get tumors that are in 100% of the progeny that inherit both Cre and SMO-M2. And these tumors are 100% fatal. by postnatal 45. We can actually change the Cree if we use GFAP Cree, which drives recombination in the brain at an earlier time point, actually in the entire brain. The only part of the brain that will get a tumor is the cerebellum. And the cerebellum will look identical to this. The tumor pathology will look like this, which is pretty close to the human tumor pathology. But the mice actually die faster. So that's kind of depicted here. We can again use GFAP Cree to drive medulloblastoma. This entire brain is going to express SMO-M2. Tumor will only happen here. With MAP1 Cree, again, get medulloblastoma. And there's a little bit of a difference, and it's important for this talk, so I'll just go over it quickly. MAP1 Cree tumors progress a little bit more slowly than GFAP Cree tumors. But if we treat with radiation, MAP1 Cree tumors are pretty responsive. We can move the Kaplan-Meier curve significantly to the right. In contrast, GFAP Cree tumors, completely radiation insensitive. With radiation, without radiation, all the mice die before they reach postnatal day 20. Okay, so MSMO and GSMO are what we abbreviate. GFAP Cree is GSMO, MAP1 Cree will be MSMO. All right, these tumors recapitulate the diversity that we find in human medulloblastoma. So we can do single-cell RNA-seq on our mouse tumors, just as we did here. And here we have 10 tumors, 5 are the GSMO variety, 5 are the MSMO variety, 20,000 cells total. We ran this through a drop-seq, sort of similar to 10x genomics, single-cell RNA-seq. And we can plot all the cells in a UMAP. So cells that are similar go into clusters together. Cells that are different from this cluster will go into a cluster over here, by group. And we can then look at the average gene expression in each cluster and identify what each cluster represents. When we do this, we can find myeloid cells, oligodendrocytes, astrocytes, fibroblasts, and a huge mass of tumor cells. They're actually arrayed according to differentiation. So the most proliferative undifferentiated cells are down here, and then the differentiated cells are at this pole. We can, because this is now in silico data, take the myeloid cells from these tumors, group them together, and recluster. And when we do that, we find actually there's 4 groups of myeloid cells that we can identify. And if we look at their gene expression patterns, we recognize that 2 of these groups are macrophages and 2 of these groups are microglia. Microglial marker CX3CR1 is expressed specifically in these 2 groups, not in these 2. CCR2, that anti-tumor macrophage marker, is expressed in one of the 2 macrophage groups. And interestingly, 3 of these 4 groups express IGF-1, the tumor-promoting hormone that's secreted by myeloid cells. And the one group that doesn't is the CCR2 positive group, completely in accord with Anna Kenny's data. So again, we have 4 groups that fit into our 4 categories. Okay, can we activate innate immune function in these tumor-associated myeloid cells and perhaps switch them from one group to another? And to do this, a couple of years ago, a couple of papers came out, one fortunately from a group that I worked with when I was at UNC, another from another group, just to show that it's a fairly widespread phenomena, that TLR7 agonists can be loaded into nanoparticles and administered in cancer models. And when we do this, we can polarize or repolarize tumor-associated myeloid cells to take on an anti-cancer effect. So I worked with the authors of this second paper, Sasha Kabanov and Marina Sokolsky, who developed a nanoparticle system for delivering drugs to the brain. It's very simple. I won't take up a lot of time with it, but roughly speaking, we dissolve drug and nanoparticles in an organic solvent, evaporate off the solvent, you get a thin film, we add water, and drug and nanoparticles will then self-assemble into my cells, where the hydrophilic part is on the outside interacting with water, hydrophobic part is on the inside interacting with the drug. if you do this, you really do get particles. We can do electron microscopy and see them. If we compared a TLR7 agonist administered either as free drug or in nanoparticles, we see improved tumor pharmacokinetics. And when we, for example, administered vismodegib, this is a sonic hedgehog inhibitor in this nanoparticle system, we found that there's a significant benefit to the nanoparticle in our tumor model compared to conventionally formulated vismodegib. And this is work we published some time ago. Okay, so we took this nanoparticle TLR7/8 agonist developed by my colleagues, and we administered it to our mice with medulloblastomas. We did this as 3 doses over a 6-day period, and we then just followed their survival. And we do this, we see actually that it actually works. The free nanoparticle is graphed in black, no treatment in blue, and either one did anything for survival. But the free rizikimod, that's the TLR7/8 agonist, in black, or no drug in blue, don't affect survival. But if we do rizikimod in the nanoparticle, we get a statistically significant increase in survival, and it slows tumor growth. How does this work? The first thing to do is simply say, what does it do to myeloid cells? If we look at the tumor in either an untreated or a treated animal, we can stain with IBA1. This is a pan-myeloid marker, and then count myeloid cells. What we find is that we actually double the number of myeloid cells in the tumor if we administer the drug. Okay, that's fine. So what does that mean? To try to unravel this, we took a 2-pronged molecular approach. We did bulk RNA-seq and we did single-cell RNA-seq. It turned out to be important to do both. While single-cell RNA-seq gave us some interesting data that I will share with you, the bulk RNA-seq also gave us data that we didn't find in the single-cell RNA-seq. And I think this is because the coverage in single-cell RNA-seq is a little bit limited. For any one gene, single-cell RNA-seq may or may not be very sensitive. It works well to detect patterns. Bulk RNA-seq, we want to know about a specific gene. We just have to sequence deeply enough to see if it's differentially expressed. So in the bulk RNA-seq, we did 4 GSMO tumors and 4 GSMO tumors untreated, the mistake on the slide, 4 GSMO tumors treated with poxrizikimod. We compared them. We found that there were 44 genes that were differentially expressed in the poxrizikimod treated tumors. Doing GSEA on these 44 genes showed that all of the top pathways that these genes conform to were immune-related, and specifically interferon signaling related. Additionally, the top 2 most strongly differentially expressed genes were a gene called Marco and another one called CD72. These are both scavenger receptor-associated proteins. And what I've shown you here is a micrograph showing Marco expression in the untreated tumor. There's basically none. These blue is DAPI stains. It shows basically these are all tumor cells. They're not expressing Marco. In the treated tumor, we see Marco positive cells throughout the tumor. But again, it's not the tumor cells. It's these cells now shown in red that are in between tumor cells. By their morphology, we can recognize them as myeloid cells. We have done a double label with IBA1, which proves that they are myeloid cells. And then interestingly, in some follow-up work, which I'm not going to show for the sake of time, wondered, well, what does Marco expression mean? So working with James Ross here at Emory and Rafi Ahmed's lab, they have a model for chronic viral infection. And we looked at what happens to mice with chronic brain viral infection. And again, we see this increase in Marco expression. So this is, I think, helpful to know that our TLR7/8 agonist is, I think, mimicking the effect of a brain virus on how the myeloid cells are going to behave. They're reacting to this TLR7/8 agonist with a big antiviral push. Okay, fine, but does that matter for tumors? Again, this just shows statistically we've increased Marco expression. What if we do single-cell RNA-seq? As I showed you before, we can get a UMAP. The tumor cells form a big cluster. And here what we've done is, again, compare For GSMO mice untreated to 4 GSMO mice treated with poxozicamod, we get a whole bunch of clusters that are different types of stromal cells, including this one, cluster 15, which is myeloid cells. And this is a projection of IBA1 expression just to show that these are myeloid cells. We can then subset the myeloid cells and ask, if you just look at this myeloid cell cluster, comparing the 4 treated and the 4 untreated samples, what genes are differentially expressed? And we get a bunch of genes that are upregulated with poxozicamod, and 2 genes that were downregulated. One of them is IGF-1, which kind of fits with what I was saying, that IGF-1 secretion is done by tumor-promoting myeloid cells, and I think we're repolarizing our myeloid cells to be tumor suppressive. And then IL-4, if anybody knows anything about IL-4, I'm all ears. I'm not really sure the significance of that. But again, we can show that we're affecting myeloid cells. And here, just to kind of prove to you that we've decreased IGF-1, we've done immunohistochemistry for IGF-1, the double label with IBA1. We can simply count the IGF-1 positive, IBA1 positive cells and show the treatment decreases IGF-1 secretion by myeloid cells in this way. Okay, we can also, since this is single-cell RNA-seq, take the myeloid set, and as I showed you before in our initial single-cell RNA-seq experiment, subcluster the myeloid cells. When we do that, instead of getting 4 clusters, we actually get now 6. And that's because the patterns of gene expression have changed and become a bit more rich. And we can look at these distribution of these 6 clusters across control or treated samples. We find that one cluster is actually only found when we treat with poxozicamod. What are these clusters? We can look at their gene expression patterns to identify them. And the key thing is we see a relative decrease in the M2 cells. If we look at the total M2 population, which is clusters 1 and 4, we see that that total population goes down. And we see induction of this group 3, this cluster number 3. And by gene expression pattern, we've identified these as dendritic cells. Summarized here, we have more M1 microglia and M1 macrophages and more dendritic cells. Okay, does this matter in terms of myeloid cell function? Their myeloid cells are supposed to phagocytose tumors, at least some of what they're supposed to do. So we wanted to look at this. Is there a good way to look at it? Our plan was we'll make tumor cells fluorescent and then treat with poxozicamod. And then we can dissociate the tumors, flow sort the myeloid cells. Myeloid cells will be fluorescent only if they engulf tumor cells. The tumor cells are going to be fluorescent. If the myeloid cells eat tumor cells, they will also be fluorescent. How do we do this? Well, it's a genetic model. We use a Cre-activated fluorescent protein bred into our model. And when we do that, we can get, if they don't have the fluorescent protein, non-fluorescent tumors. Or if they do have the fluorescent protein, tumors in the cerebellum that fluoresce. So we took our mice with fluorescent medulloblastomas. We treated them with poxozicamod, dissociated the tumors, and then did flow cytometry with CD45 and CD11b to identify myeloid and microglia and macrophages. And we can then quantify GFP fluorescence in the myeloid populations. We can separate the macrophage population and the microglial population. And if we simply graph how many cells are GFP positive and treated and untreated, we see a big increase in the GFP positive myeloid cells with the drug. So kind of quickly summarize, the drug works. Poxozicamod induces prolonged survival in the mice. It has to be done as a nanoparticle in order for it to work. And we're engaging the immune system in a way that upregulates Marco and scavenger receptors and downregulates IGF-1. This increases phagocytosis of tumor cells, and it expands the dendritic population. This last thing is important because it's going to segue into the next thing I want to talk about, and I promise I'll go fast, that there's a potential link to adaptive immunotherapy. Okay, so how could we move this forward for patients? Our drug extends survival in mice with medulloblastoma. That's fine. But as you see, none of the mice are actually cured. And you could say, well, this Kaplan-Meier curve does go out. But if we dissect these animals, even at P45, we find that there's still residual tumor there. If we waited long enough, I'm quite sure it would eventually progress and kill the animal. And this is not surprising. There's no drug in medulloblastoma therapy that's used as a single agent. There's no single agent that's effective. So what could we combine this drug with? And one thought is to combine it with immunotherapy, other immunotherapy. So what would be the rationale for combining with T-cell mediated therapy? As I said, stimulating the myeloid cells to fight the tumor works, but it's not curative. In leukemia and lymphoma, CAR T cells, these are T cells genetically engineered to attack the tumor, can be curative. CAR T cells have not yet been shown to be effective and certainly not curative in brain tumors. It's been more complicated to use CAR T cells in solid tumors. One consideration is that myeloid cells in the brain may inhibit T cell function. My colleague at Duke, Peter Fecci, has published extensively on the idea of T cell exhaustion in patients with glioblastoma. And when we stain our tumors for T cell markers or just even CD3 for pan lymphocyte markers, we find almost no CD3 positive cells in our tumors. So T cells are going to be rare, but perhaps they could be added. I guess the absence of T cells, I think, speaks to the fact that tumor microenvironment is not conducive to them. But again, perhaps they could be added, and perhaps by altering the microenvironment with our poxrozicamod, we could make the T cell environment, make the tumor microenvironment more T cell conducive. Okay, so can we model CAR T cells in our mice? What are CAR T cells in brief? This is, imagine a T cell. The CAR is a chimeric antigen receptor. It's a genetically altered T cell receptor that's been modified with an antibody domain that binds to a target on the tumor. The target I want to talk about is B7H3. B7H3 is a protein expressed in almost all pediatric brain tumors, not expressed in the normal brain. So it seems like an ideal target for CAR T cell therapy. T cells can be harvested from a donor mouse, expanded in culture, transduced to express B7H3 CAR. And this is work by my colleague, John Doty at UNC. They used a mouse glioma xenograft model. They put human cells, human tumor cells into an immunosuppressed mouse, and they then treated the mouse with mouse T cells engineered to express a CAR that recognizes human B7H3. And doing this, they were able to get a nice survival benefit in their model. But recognize if you put human GBM cells into a normal mouse, it will also clear the tumor very quickly. So we have to start out with an immunocompromised mouse. We then add T cells and we get rid of the tumor. All we're kind of doing is recapitulating the normal immune function of the mouse. An important test for CAR T cells is that we should be able to put CAR T cells into a mouse that's immunocompetent and clear a tumor that survives in an immunocompetent mouse. Okay, so could we take our mouse model, which is an immunocompetent model, and do CAR T cell therapy? Not easily, because this model turns out not to express B7H3. So this is one of the tumors from one of our mice. We've stained for B7H3 in red, and you see there's no red. It's a little red here in the choroid plexus, but the tumor is not expressing B7H3. So to address that, we made a new transgenic mouse that, like our SMOM2 mouse, has the lock-stop locks. It then has the coding region for B7H3. The gene is called CD276. We also added luciferase just to make it a little easier to do some of the work. And with this new transgenic mouse, we get tumors that express B7H3 nicely. They also express luciferase that increases over time so that we can follow tumor growth. So to validate this, we made slice cultures from our mouse with the tumor. And again, you can see the tumor here is bioluminescence from luciferase. And what we did is we added CAR T cells to these slice cultures. We compared either no cells, non-transduced T cells, or CAR T cells. We did this on day 0. We then on day 2 looked at luciferase expression. What we see is that either no T cells or control T cells don't change luciferase expression. Expressed here as a percent of the amount you had at day 0, the amount of luciferase you have at day 2 goes up slightly. On the other hand, if we add CAR T cells, luciferase goes down, providing some evidence that these CAR T cells, which were engineered against human B7H3, do in fact work against mouse B7H3 in our model. We also did ELISA for IL-2, and it shows that the CAR T cells on day 1 are secreting IL-2 into the medium when we add them to our slice cultures. In contrast, slices treated with either saline or untreated don't secrete IL-2. Okay, so great. Now we have a primary animal model that expresses B7H3, an immunocompetent host that gets these nice heterogeneous tumors. What happens if we treat with CAR T cells? So here are just some representative images to show you what we see if we look at luciferase. Without CAR T cells, the animals progress from tumors bigger by P10. The tumor is actually, luciferase wanes, but the animal dies. Basically, we can see that there's a tumor there, and it stays there, and the animal dies. If we treat with CAR T cells, we tried 2 different doses. We found we have to do this by intracranial injection. We inject CAR T cells directly into the brain, and quickly the amount of tumor decreases. We do the CAR T cell injection at P4. By postnatal day 10, there's less tumor, and with a higher dose, we really do keep the tumor suppressed over time. Where do CAR T cells go when we inject them? So these are transgenic cells, so we can transduce them with a marker that we can then stain for. So here we have HA tagged CAR T cells. We injected them ICV at postnatal day 4 and then sacrifice the animal postnatal day 10. Where are the CAR T cells? They're in red. You can see they're all over the brain, but they are actually here in the tumor as well. If we look at high magnification, we can see the CAR T cells will inject it into the ventricle, migrate into the tumor. They will penetrate a solid tumor, and they'll do whatever they'll do. And one of the things they'll do is slow tumor growth. As we show here, with either smaller dose of CAR T cells or bigger dose of CAR T cells, we can drive the Kaplan-Meier curve to the right. Now, just like I showed you with, well, let's just quickly summarize them. So we can make a model that expresses B7H3. The same CAR that recognizes human B7H3 will recognize mouse B7H3. This is important because if we ever wanted to go forward with this, we want to see it happen in parallel to do studies in mice and to be able to do studies in patients. The CAR T cells, as you saw, will home to the tumor after ICV injection. And lastly, they show efficacy and recurrence. narrow particle. Yeah. Yeah, ex vivo. You know, once you inject them into the animal, it's actually quite difficult. I think that's the one thing we having old, tiny boys, put it on a python and then inject the mice and then these are live python. Yeah. But that's the without those kind of having. Yeah, so yes, I agree. That's a really good technology. We should talk about that. What we've done previously is we've used NMR to detect the nanoparticle carrier. It's hard. We found it hard, at least, to detect the carrier by mass spec, but by NMR we can detect the carrier and the drug separately, and then look at whether the carrier and the drug stay together in the tissues. What we found is that, at least for vismodegib, when we had that in the nanoparticle would separate from the drug at the tumor. So it seemed to stay together in the blood, but then in the tumor, the drug would get in and the nanoparticle would not. What we think is that for one reason or another, the nanoparticle seems to release the payload at the blood-brain barrier. But that's a whole other conversation. Thank you. I appreciate the question. Just to kind of wrap this up, what I want to describe is a kind of concept for harmonized therapy where we are treating both the innate immune environment and treating through adaptive immunotherapy. Great. So the concept would be poxozygomod conditions the tumor microenvironment, makes the microenvironment more supportive for CAR T cell therapy. It'll be more of a pro-inflammatory environment. And at the same time, CAR T cells can remove refractory tumor cells that are growing through the CAR T cell treatment, through the poxozygomod treatment. And CAR T cell therapy may be optimized by redosing. We've only played around with a single dose. So the concept would be that this combination together may allow us to make progress in solid tumor CAR T cell therapy and brain tumor CAR T cell therapy in a way that we haven't already done. All right, so with that, I want to acknowledge people in my lab, people in my past team at UNC. I have some really great Emory collaborators, James Ross, whom I mentioned. Trent Spencer has been helping me re-envision the concept of CAR T cell therapies, and I didn't have time to talk about it today, but brought up a very interesting idea of using biallelic T cell engagers rather than CARs, as a sort of different way of bringing T cells to the tumor. Nick Barbell and Ray Dingledine have shared with me their CCR2 RFP mice, which we have bred into the tumor line, and these are going to allow us to look more closely at what poxozygomod is actually doing to the CCR2 positive population. Again, my collaborators at UNC, Drs. Sokolsky and Kabanov, and Doty, and my funding sources. So with that, thanks everybody.
Okay, thank you, Dr. Gershon. If you have any questions, please use the Q&A feature located at the bottom of your screen. And while we wait on questions, I wanted to mention that next week there is no grand rounds. Instead, there is the internal critical updates in clinical research. The week after that, on October 4th, we will have Emory's Dr. Saurabh Chawla presenting pancreatic cancer screening, moving the needle, or a wild goose chase. To view all upcoming Winship Grand Rounds lectures, please visit our Grand Rounds page on the Winship Cancer Center website or the Winship calendar. Do we have any questions from the room?